Wait, don't come up yet. Is Helena Sverak from Dr. Natalie Stranatka's lab?
So thank you so much for the opportunity to speak here today. I'm excited to share my work about inhibition of Klebsiella pneumoniae carbapenemase, and with that I will jump right in. So antibiotic resistance is kind of a growing problem as we all know it, and the recent data on the burden of resistance is that it's now the third leading cause of death globally from 2022 meta-analysis by the Lancet. But unfortunately, there's not much financial incentive by companies to look into the research. So our group looks at studying proteins and mechanisms that are involved in resistance and pathogenicity in these multi-drug resistant infections. Now, the World Health Organization has put together a list of the most concerning pathogens for resistance, which are deemed the escape pathogens. And many of these share different resistance mechanisms. And of that statistic that I talked about, of it being the third leading cause of death, 70% of those deaths are due to resistance in beta-lactams and fluoroquinolones. So I'm going to be focusing the rest of this talk on beta-lactam resistance. Now, beta-lactams are a type of antibiotic that work by mimicking the substrate in cell wall synthesis. And beta, the largest resistance factor against them, or resistance mechanism against them in gram-negative bacteria, are beta-lactamases. And these are enzymes that go in and they cleave this structure that's critical to the drug, which is this four-membered ring, and it renders them inactive. And there's now a subset of beta-lactamases that have developed to cleave carbapenems, which were originally a beta-lactam of last resort. And these enzymes are carbapenemases. And in Klebsiella pneumonia, a carbapenemase was discovered recently that's become kind of wildly successful, and it's called Klebsiella pneumonia carbapenemase 2, KPC2, as I use as an acronym. And it's been so good that it's spread into the other escape pathogens as well. And in bloodstream infections of patients with Klebsiella pneumonia that have this carbapenemase, fatality initially was up to like 50%. So we're very interested in inhibiting this enzyme. And so our collaborators in the Baker lab used computational design to create this kind of new type of inhibitor with cyclic macropeptides. And they'd previously used this technology to create an inhibitor against another concerning type of beta-lactamase, the NDM1 metallo-beta-lactamase. And so they turned and applied this technology to KPC2 to create a mechanistic-based inhibitor that kind of mimics the transition state in the active site and uses a boronate in kind of a key position to stabilize some charges and hopefully create a tight binder. And so it's been my project to take 4 of these different designed inhibitors and to characterize their inhibition of KPC2 both kinetically as well as to try to structurally characterize them using X-ray crystallography to be able to see atomic resolution of their binding. And to do this, I used kind of canonical protein purification. So I transgenically expressed KPC2 in E. coli. I took the cells, lysed them, and then went through various steps of protein purification and chromatography to get a clean sample. And then for the kinetic assays, I use a beta-lactam analog, which is called nitrocephin, and is pictured up there turning yellow to red. And so it's the same structure as a beta-lactam, but it changes color. And so we're able to use that kind of in competition with these inhibitors to see the effectiveness of these inhibitors in preventing KPC2 from cleaving a beta-lactam. And then in order to to be able to analyze it with crystallography, you take very high concentrations of protein, and then you expose it in lots of different conditions to hopefully find a condition in which it's able to crystallize. And then you take your crystal and you bring it to a synchrotron, where if you shoot it with an X-ray, you can then take the diffraction pattern and solve a model of structure. And so I was able to do this with all 4 of the inhibitors and characterize a range of different affinities of inhibition between the 4 inhibitors that we had, going from 50 nanomolar to 50 micromolar affinity, or IC50. And we're hopeful, and then we're able to achieve 2.2 or better angstrom resolution in all of the structures, which allows us to see the interactions of the specific side chains and how they kind of are binding in the active site. And this type of data, we hope, is going to be useful for development of kind of future antibiotics and inhibitors of these types of enzymes, and hopefully be kind of a tool towards fighting multi-drug resistant bacteria going forward. So with that, thanks for listening.
I'm sorry, have you ever tried the linear version of that cyclic peptide as a control?
We were specifically looking at the cyclic peptides, but they most likely wouldn't work in the sense that our biggest difference in affinity was between 1B and 9B, which actually have the exact same sequence but are only cyclized in a slightly different area. And so if they weren't cyclized at all, they wouldn't bind at all, most likely.
Go ahead, Isaac.
That's excellent work. Have you considered looking at AI and machine learning to try to optimize these? And I only say that because Jonathan Stokes from from McMaster published a paper recently on literally discovering a brand new antibiotic. I can't remember for what, but just by using AI and machine learning. Really powerful tool.
Absolutely. So the program that our collaborators use, Rosetta Commons, I'm not completely familiar with it, but I would be surprised if it didn't use some sort of machine learning or additional kind of current technologies. It's one of the leading softwares in the field, and AI has kind of been an incredibly helpful tool for structural biochemists with the invention of AlphaFold. So yeah, we heavily use that in our work.
Amazing, thanks.
Do you know if the lactam is susceptible for protease activities? I mean, it's an amide bond. Can it be cleaved by proteases, do you think?
Sorry, can you
So
repeat?
lactam, right? It's an amide bond in the ring.
Yeah.
So is it susceptible for proteolytic activities? Can it be cleaved by a protease?
Beta lactams?
Yeah. Mm-hmm.
I don't think that, I think that regular antibiotics are relatively protected from protease activity, but I can get back to you for sure. I don't think so.
Great, thanks.